Hello everyone, welcome to Tawhid Foundation for Allied Healthcare Sciences, Bangalore. So today's class is the continuation of antigen antibody reaction, part 3 of immunology for BSc MLT third year students. So in last class, we ended in last class till single diffusion in one dimension. Will continue from here now. So precipitation in gel, single diffusion in one dimension. In this setup, you can see rectangular gel block or stripe. Inside the gel, the antibody is uniformly distributed throughout the entire gel, act as a reservoir of antibody. So on the upper surface or at one end of the gel, there's a narrow trough or well cut into it, and that trough is filled with the antigen solution. Now, look at the arrows. They show the antigen molecule diffusing downward and sideways into the antibody containing gel. As this antigen move through the gel, they gradually encounter increasing concentration of antibody. At a certain distance from the trough, the concentration of antigen and antibody become exactly equal, known as the zone of equivalence. At that region, they form insoluble antigen antibody complexes that appear as a thin visible precipitin line running parallel to the trough. So that's the precipitation band. The important concept here is that only one component, the antigen, diffuses through the gel while the antibody remains stationary. Hence, we call it single diffusion. This method is primarily used to compare antigen concentration. The distance of the precipitin line from the trough is proportional to how much antigen was initially placed. Higher antigen concentration result in the line forming further into the gel because the antigen has to diffuse further before reaching equivalence. So in short, this diagram demonstrates that precipitation occurs at the point of antigen-antibody equivalence, and the position of this line can be used to estimate the concentration of antigen. Next is double diffusion in one dimension, Oakley procedure. In the Oakley procedure, both the antigen and antibody are allowed to diffuse toward each other through a layer of plain agar, allowing the reaction to occur in one dimension. A test tube is prepared with three layers. The bottom layer contains agar with the antibody. The middle layer is plain agar that serve as a diffusion medium, and the top layer holds the antigen solution. As diffusion proceeds, the antigen moves downward and the antibody moves upward through the plain agar layer, where the two meet in optimal proportions. A precipitin band forms within the middle layer. These tests demonstrate antigen-antibody diffusion and interaction in a clear and measurable way. The Oakley method is more refined than the Audin technique and can show multiple bands if several antigenic components are present. So as you can see in this image, now this diagram shows a different setup, that is double diffusion in one dimension. Here, instead of one component diffusing, both the antigen and antibody diffuse towards each other in a linear one-dimensional arrangement. If you look at the figure, you'll see a gel block with two parallel troughs or slots cut into it, one on the top side and one on the bottom side. The upper troughs contain antigen solution and the lower troughs contain antibody solution. When the experiment starts, both antigen and antibody molecules begin to diffuse through the gel. The antigen diffuses downwards and the antibody diffuses upwards. As they move towards each other, their concentrations change gradually, high near their own trough and decreasing as they move away. In the central region, their concentrations become equal. As you can see in this image, this is again the zone of equivalence. At the exact zone, antigen antibody complexes form and become insoluble, creating a sharp white precipitin line between the two trough. As you can see, this sharp white precipitin line. So that's the visible evidence of their reaction. This is called double diffusion because both the reactants are mobile and one dimension, because the diffusion occurs only along a straight line between the two troughs. This method is useful for comparing multiple antigens or antibody samples. If you place several antigen stuffs opposite a single antibody trough, each will form its own precipitin line depending on antigen concentration and diffusion rate. So the key difference from the previous slide is that in single diffusion only the antigen moves, as you can see in this image. In single diffusion only antigen will move, but only the antigen move through a stationary antibody gel, while here both the antigen and antibody move and meet in the middle to form the precipitate. Next is single diffusion in two dimensions. In the radial immunodiffusion method, also known as the Mancini technique, the antibody is uniformly mixed within an agar layer poured on a slide or petri dish. Small circular wells are cut into gel and each well is filled with an antigen solution of known or unknown concentration. As the antigen diffuses radially outward in all directions through the gel, it reacts with the antibody and forms a circular precipitin ring. So, the diameter of the ring increases until equilibrium is reached and is directly proportional to the antigen concentration. By comparing the ring diameter of unknown samples with those of known standards, the exact concentration of antigen can be determined. This method is highly reliable for quantitative estimation of serum proteins and immunoglobulins. This is the image. Here the image shows a series of wells with known antigen concentrations forming rings of increasing diameter. The graph beside it plots the square of the ring diameter against the square of the ring diameter against antigen concentration, showing a straight line which helps in preparing a standard curve. Unknown sample can then be read by comparing their ring diameter to this line. The diagram helps students connect visual precipitin rings with quantitative measurements. Next is double diffusion in two dimension, Auchterlonie technique. So the Auchterlonie double diffusion technique is one of the most widely used immunodiffusion methods in two dimensions. Agar gel is poured onto a glass plate and several small wells are cut into it. The central well contains the antibody and the surrounding wells contain different antigens. So both antigen and antibody diffuse radially down towards each other through the gel. When they meet in optimal proportion, visible precipitate lines forms between the wells. The pattern of these lines provides information about the relationship between different antigens. Identity fusion, that is when two antigens are identical, the lines fuse into one smooth arc. Non-identity, that is crossing, when they are unrelated, the lines cross each other. Third is partial identity, that is spur. When they share some epitopes, a spur forms indicating common and unique determinants. This test is extremely useful for comparing antigenic relationship, identifying unknown antigens, and studying antibody specificity in research and diagnostics. This is the Auchterlonie technique. In this test, both antigen and antibody diffuse through agar gel and form visible lines of precipitation where they meet in optimal proportions. This diagram shows three outcomes depending on how the antigenic structures are related. So, reaction of partial identity, this left side of the image, in the first part of this diagram, you can see three wells. So, the upper well central contains the antibody, while the other two wells contain antigen A and antigen A1. So, Notice that the precipitation lines from both antigen fuse together at first, but then form a small spur or projection pointing towards antigen A. This spur indicates that antigen A1 has all the epitopes present in antigen A plus some additional unique epitopes that are not shared. So the antibodies react with the shared epitopes to form the common fused lines, and the spur represents the extra determinants unique to A1. This pattern is called partial identity because the two antigens share some but not all antigenic determinants. In short, lines merge plus spur formation is equals to partial identity. So next is a reaction of non-identity, that is this middle of the image in the second part of this diagram. Again, the top well contains antibody and the two lower wells contain antigen A and antigen B. This time the precipitin line forms both antigen cross each other. They do not merge or fuse. This crossing pattern indicate that two antigens are completely unrelated, that is A and B. They have no common epitopes. Each antigen reacts independently with its specific antibody molecules, and therefore two separate non-interacting lines appear that simply intersect. So lines cross each other is equals to non-identity, means the two antigens are antigenically different and unrelated. Third is reaction of identity. So the antibody well is again at the top, and the two lower wells both contain antigen A, that is identical antigens. Now look at the lines. Two precipitin lines from both antigen fuse completely. As you can see, fuse both this line fuse completely, and so this smooth arc shows that both antigenic well contain the same antigenic structure recognized identically by the antibody. Since the antibody reacts with both in exactly the same way, the reaction zones merge perfectly. So single fuse line is equals to reaction of identity. It indicates the antigens are identical, that is same epitopes, same antigens, that is A. Next is application of radial immunodiffusion. Radial immunodiffusion has a wide range of clinical and diagnostic applications. It is used for quantitative estimation of immunoglobulins such as IgG, IgM, and IgA in serum, helping in the diagnosis of immunodeficiency or hypergammaglobulinemia. So it is also employed for screening sera for antibodies to influenza viruses, aiding in epidemiological studies and vaccine response monitoring. Another important use is the determination of serum complement, that is component C3, which is vital for assessing immune system function. This technique also measures transferrin levels, which are important in the evaluation of anemia and nutritional statuses. In obstetric diagnosis, of fetoprotein levels in maternal serum are estimated by this method to detect fetal abnormalities. So, like natural tube defects, overall radial immunodiffusion remains a simple yet powerful quantitative immunological assay. Next is immunoelectrophoresis. Immunoelectrophoresis is a powerful technique that combines electrophoresis and immunodiffusion to analyze mixtures containing multiple antigens. First, the serum or antigen mix is placed in an agar gel and subjected to electrophoresis under the influence of an electric current. So, different antigen molecules migrate through the gel at different rates based on their molecular size or electrical charge. Once the separation is complete, a trough is cut parallel to the electrophoretic line, and antiserum containing the corresponding antibodies is added. So the antibodies then diffuse laterally through the gel towards the separated antigen bands. As the antigen and antibody fronts meet at optimal proportions, precipitin arcs form, each arc representing a distinct antigen antibody reaction. Thus, immunoelectrophoresis not only separates but also identifies multiple antigens in a single sample, giving both qualitative and semi-quantitative information about serum proteins and other components. This immunoelectrophoresis here in A part, antigens placed in an agar gel are separated by means of an electric current. Positively charged molecules are drawn towards the negative pole and negatively charged ones move towards the positive pole. So second part is or trough is cut into the agar between the wells and filled with antibody. See, third part, it is curved precipitin band forms where antigen and antibodies diffuse to meet and react. So this is the schematic representation of electrophoresis, which is important for exam purpose. Next is electrophoresis application. Electrophoresis is a routine diagnostic and research tool for studying serum proteins. It helps in the detection and quantification of albumin and globulin, various globulins, allowing clinicians to evaluate protein abnormalities. One major use in the diagnosis of multiple myeloma, where abnormal immunoglobulin bands, that is M protein, appears as distinct additional arcs. The technique also helps to differentiate antigenic variants in microorganisms. Identifying closely related species or strains based on antigenic profiles. Furthermore, it is applied in the analysis of autoimmune disorders or and immunodeficiencies where changes in immunoglobulin levels or absence of specific protein arcs can be clearly visualized. Overall, immunoelectrophoresis provides a visual fingerprint of the body's serum proteins, essential for disease diagnosis and immunological research. Next is countercurrent immunoelectrophoresis, that is CIEP. Countercurrent immunoelectrophoresis is a rapid and sensitive modification of immunoelectrophoresis. In this method, the antigen and antibody are placed in opposing wells cut into an agar gel. When an electric field is applied, the antigen and antibody molecules migrate towards each other. The antigen usually moving towards the anode and the antibody towards the cathode. As they meet in the gel, they form a precipitin line between the wells within a short period, often less than 1 hour, because the electric current accelerates molecular movement. This technique provides faster results than traditional diffusion methods and enhances reaction sensitivity. CIEP is especially useful for detecting bacterial or viral antigens such as hepatitis B, surface antigen, that is HBs, AG, alpha fetoprotein and cryptococcal antigens in clinical samples like serum or cerebrospinal fluid. This diagram shows an agar plate with two wells, one containing antigen and the other containing antibody. Placed opposite each other, when current is applied, the antigen migrates towards the anode and the antibody towards the cathode. In the middle, a sharp white line appears where they meet, forming a precipitin band. The arrows in the figure illustrate opposite migration directions. Hence, the term countercurrent emphasizes that electric current accelerates diffusion, giving faster results compared to passive diffusion method. Next is rocket electrophoresis by Laurel technique. Rocket electrophoresis, also called the Laurel method, is a quantitative immunoelectrophoretic technique used for antigenic estimation. In this method, the antibody is uniformly incorporated into the agar gel and small wells are punched to hold antigen solutions of known or unknown concentration. When an electric field is applied, the antigens migrate through the gel and react with the antibody to form rocket-shaped precipitate peaks. The height of each of rocket is directly proportional to the concentration of antigen in the sample. By comparing the height of unknown samples to those of standards, precise quantification is achieved. This technique is rapid, reproducible, and widely used for measuring serum proteins and hormones, as well as for standardization in diagnostic assays. Rocket electrophoresis combines immunodiffusion with electrophoresis. The antigen migrate through antibody containing gel and form rocket-shaped precipitation zone. The height of each rocket corresponds directly to antigen concentration, making it a quick quantitative immunological technique. So, as you can see in this figure, in this figure, rocket electrophoresis combines with immunodiffusion with electrophoresis. This is also important for exam purpose. Next is radial immunodiffusion through Mancini method. So in this antibody in gel, antigen diffuses radially from wells. Precipitin ring that is diameter is proportional to antigenic concentration, and it allows quantification of antigen, and it is useful for serum protein measurement. In this diagram, here as I mentioned this earlier, next is agglutination reaction. So agglutination reaction is an antigen-antibody reaction where particulate antigens such as cells, bacteria, or latex particles clump visibly when mixed with their specific antibodies. This reaction occurs because the antibodies cross-link the surface antigen on different particles, forming a lattice network that become visible as clumps. For agglutination to occur, the antigen must be particulate and the antibody bivalent, allowing effective binding. So, agglutination is a cornerstone of diagnostic immunology, forming the basis for many serological tests such as blood typing, bacterial identification, and antibody detection. It is a quick, visual, and highly specific reaction used both qualitatively and quantitatively in laboratories. Next is direct agglutination. So in direct agglutination, natural particulate antigens react directly with their corresponding antibodies without any modification. Common examples include red blood cells and bacteria, which naturally carry antigens on their surfaces. When mixed with specific antibodies, visible clumping occurs as antigen-antibody complexes form. This reaction is used for blood grouping, bacterial identification, and other tests where the antigen exists naturally on cell surfaces. It is simple, requires minimum reagents, and gives a rapid visual confirmation of antigen-antibody interaction. So now let's look at this diagram showing the direct agglutination test, which is one of the simplest and most common serological methods used to detect antibodies against particulate antigens like red blood cell, bacteria, or latex particles. So principle is straightforward. When a specific antibody reacts with its corresponding particular antigen, the antibody binds to multiple antigen particles at once, forming visible clumps or aggregates called agglutinates. If no corresponding antibody is present, the particles remain separate and no visible clumping occurs. So, look at the part A of this figure. This shows a positive agglutination reaction. Here, the well or tube contain the antigen suspension mixed with the patient's serum, which may contain antibodies. In this reaction, there are sufficient antibodies present in the serum to bind to the surface of multiple antigen particles at once. So each antibody molecules with its two binding sites acts like a bridge, linking antigen together and forming a large lattice network of antigen antibody complexes. In this diagram, you can see that this result in a loose mat or a carpet-like layer or clumped particles spread over the bottom of the well. This spreading indicates successful agglutination, meaning the antibody is present and has reacted with this antigen. So in short, the antibodies have cross-linked the antigens. A visible mat forms on the well bottom. This is interpreted as a positive test result showing the serum contains specific antibodies against the antigen. Exam Examples of direct agglutination. So, direct agglutination is widely used in clinical diagnosis. The Vidal test detects antibodies against Salmonella typhi and Salmonella paratyphi, Salmonella species, for diagnosing typhoid fever. Brucella against agglutination test helps confirm brucellosis infection by detecting antibodies in patient serum. The Wheel Felix test identifies rickettsial infections by demonstrating agglutination between patient serum and protease stains sharing cross-reacting antigens. Blood grouping In blood grouping, agglutination reaction determines ABO and Rh blood types by mixing RBCs with specific antisera. All these tests rely on visible clumping as a sign of specific antigen antibody reaction. Next is tube agglutination test. The tube agglutination test is a quantitative method to measure antibody levels in serum. A series of serial dilutions of patient serum is prepared in separate test tubes. A standard suspension of antigen is then added to each tube and the mixtures are incubated under controlled conditions. After incubation, tubes are examined for visible clumping or sedimentation of antigen antibody complexes. The endpoint titer is the highest serum dilution showing visible agglutination representing the concentration of antibodies in the sample. So these tests provides valuable quantitative information for diagnosing disease such as typhoid, brucellosis and others. Next is indirect passive agglutination. So, principle behind this, in indirect or passive agglutination, soluble antigens that normally do not cause visible clumping are attached to carrier particles such as latex beads, bentonite particles or red blood cells. When the coated particles are mixed with specific antibodies, agglutination occurs, indicating the presence of the corresponding antibody. This modification increases test sensitivity, allowing detection of very small quantities of antibody or antigen. Passive agglutination is especially useful when the natural antigen is not particulate or poorly reactive, extending the range of agglutination-based diagnostics. So in this indirect agglutination test, these two diagrams, they show how the indirect agglutination test works in two different setups. In both the cases, instead of using whole bacterial cells or red blood cells as natural antigens, we artificially attach antigens or antibodies to inert carrier particles like latex beads or charcoal particles or bentonite. These tiny particles act as visible platforms to display antigens or antibodies and make the reaction easier to see. So, principle of this indirect agglutination, the principle is that when antigen or antibody is coated onto particle, it behaves like a large visible antigenic complex. If the corresponding antibody or antigen is present in the test sample, it binds to these coated particles and causes them to clump together, forming visible agglutination. So in this figure A, Here the latex particles are coated with specific antigens. As you can see that the surfaces of the spherical particles carry the antigen molecules, the circle one. When this antigen coated latex suspension is mixed with serum sample containing specific antibodies like IgM antibodies, the antibodies bind to the antigen on the particle surfaces. So in short, latex beads are coated with antigen. If antibody is present, visible agglutination is seen. This is a positive result indicating the presence of antibody in the serum. In this figure P, which shows the opposite arrangement, here the latest particles are coated with specific monoclonal antibodies instead of antigens. When this suspension is mixed with a test sample containing antigen, the antigen binds to the antibodies on the particular surfaces. So each antigen molecule can attach to multiple antibody coated beads, causing the beads to cross-link and form visible clumps. This is again a positive agglutination reaction. So in short, latex beads are coated with antibody. If antigen is present, visible agglutination is seen. This is positive result indicating the presence of antigen in the sample. Next is applications of passive agglutination. Passive agglutination has numerous clinical applications. The C-reactive protein, that is CRP latex test, detects the inflammation markers in serum. Detection of rheumatoid factor, that is RF factor, in rheumatoid arthritis is done using latest particle agglutination and is also applied in viral antigen or antibody detection such as in HIV and hepatitis B virus testing. Certain hormones and drugs can be detected or quantified by passive agglutination formats because of its high sensitivity and simplicity. This technique is widely used in modern serological testing. Next is hemagglutination principle and example. Hemagglutination involves the clumping of red blood cells RBCs when exposed to specific antibodies or certain viruses. In this reaction, antibodies recognize and bind to antigens present on the surface of RBCs, cross-linking them to form visible clumps. This principle forms the basis for blood grouping and cross-matching, essential in transfusion medicine. Many viruses, such as influenza and mumps, possess surface proteins capable of directly agglutinating RBCs, a property exploited in laboratory diagnosis and research. Hence, hemagglutination serves as a valuable tool in both virology and immunohematology. Next is in this image, as you can see, red blood cell and hemagglutination. So this red blood cells plus virus give rise to hemagglutination. Next is viral hemagglutination and inhibition. So viral hemagglutination such as influenza, certain viruses including influenza and mumps naturally agglutinate red blood cells due to the presence of hemagglutinin proteins on their surfaces. So in viral hemagglutinin test, the presence of the virus is confirmed by the clumping pattern of RBCs. As you can see, in contrast, the hemagglutination inhibition, that is HI test, detects antiviral antibodies in the patient's serum. If the patient's serum contains specific antibodies, these will bind to viral particles and block hemagglutination, preventing RBC clumping. This inhibition indicates prior exposure or immunity to that virus. The HI test is widely used for vaccine evaluation and epidemiological surveillance of viral diseases like influenza. So, in this diagram, showing viral hemagglutination, HA, which is a method used to detect viruses that can bind to and agglutinate red blood cells, that is RBCs, such as influenza virus. So the principle is simple. Certain viruses have surface proteins called hemagglutinins that can attach to receptors on red blood cells. When enough virus particles are present, they cross-link RBCs, forming a lattice that prevents the RBCs from setting at the bottom of the well. So structure of this diagram, microtiter plate is arranged, each containing a mixture of red blood cell and virus at different dilution, then serial dilution of virus, and then red blood cell setting patterns. So positive hemagglutination, the virus has cross-linked RBCs forming a diffuse mat across the well. The RBCs will not settle at the bottom, so the well appears cloudly. So this diagram you can draw for hemagglutination question. Next is Coombs test. Coombs test, direct and indirect. The Coombs test, also known as the antiglobulin test, detects antibodies that react with red blood cells. So in direct Coombs test, red cells taken directly from the patients are tested to detect antibodies or complement components that are already bound to the RBC surface, a process known as in vivo sensitization. In indirect Coombs test, the patient's serum is first incubated with reagent red cells. If antibodies are present in the serum, they attach to red cells in vitro. After washing, anti-human globulin reagent, that is Coombs reagent, is added, which link the bound antibodies and produces visible agglutination. This test is fundamental in transfusion medicine for detecting weak or incomplete antibodies that may cause hemolytic reactions. In this diagram, so the Coombs test, also called as the antiglobulin test, is designed to detect antibodies bound to red blood cells, that is RBCs, that are not able to produce visible agglutination on their own. It is a very important test in blood typing, detecting hemolytic disease of the newborn and autoimmune hemolytic anemia. So as you can see, it is divided into two parts, A and B. So here in A part, red blood cells are mixed with anti-RH antibodies. If the RBCs carry RH antigens, these antibodies will bind to the so these antibodies will bind to the antigens on the RBC surface. However, the number of bound antibodies is too small to cross-link the RBCs and form visible clumps. So even though antibodies have bound to the RBCs, no hemagglutination is seen at this stage. This is why we cannot detect RH positivity with just the first antibody reaction. When antibody concentration is low, the RBCs appear free and separate. In part B, to detect these bound but non-agglutinating antibodies, we add anti-human globulin antibodies, that is Coombs reagent, which are usually prepared in rabbits. These anti-human antibodies recognize the FC portion of human IgG antibodies already bound to the RBCs. When the Coombs reagent bind to the human IgG on multiple RBCs, it cross-links the RBCs, forming visible agglutination. This visible clumping confirms that the RBCs carries the RH antigens and that anti-RH antibodies were bound, making the person RH positive. Applications of Coombs test. The Coombs test has several important diagnostic applications. It is used to detect erythroblastosis fetalis, also called hemolytic disease of the newborn, caused by maternal antibodies attacking the fetal RBCs. In blood transfusion compatibility testing, it ensures that donor and recipient blood are immunologically compatible. It also aids in diagnosing autoimmune hemolytic anemia, where the patient's own antibodies destroy the RBCs. Additionally, the test can investigate drug-induced hemolysis, where certain medication can cause antibodies to form against red cells. Thus, Coombs test plays a vital role in hematology and transfusion safety. Next is Rose-Waller test and coagulation. The Rose-Waller test is a classical agglutination test for rheumatoid arthritis. In this method, sheep red blood cells are coated with rabbit IgG, and when mixed with serum containing rheumatic factor, agglutination occurs, confirming the presence of the antibody. Coagglutination uses bacteria such as Staphylococcus aureus coated with specific antibodies. When these coated bacteria are mixed with corresponding antigen, visible clumping occurs, indicating a positive reaction. This technique increases sensitivity and specificity of bacterial antigen detection and is commonly used for staphylococcal antigen identification and rapid bacterial diagnostics. So coagulation is a variation of the agglutination test in which antibodies are bound to the surface of bacteria instead of being free in solution. The most commonly used bacteria is Staphylococcus aureus COWAN1 strain, which has protein A on its cell wall. Protein A binds to the Fc region of antibodies, leaving the Fab region free to recognize antigen. So again, this image is divided into two parts, A and B. Part A, here the S aureus cells are coated with specific antibodies. Protein A on the bacterial surface binds the Fc portion of the antibody, orienting the antibodies in such a way that their antigen binding sites, that is Fab regions, point outwards. These antibody-coated bacteria now act as a visible carriers like tiny markers which can interact with specific antigens in a test sample. So the antibodies are immobilized on the bacteria. So the bacteria themselves become part of the visible lattice with agglutination occurs. When agglutination occurs. In part B, when antibody-coated S. aureus is mixed with a sample containing the desired antigen, the antigen binds to the Fab portions of the antibodies on different bacteria. These binding cross-links multiple bacteria via the antigen, forming a lattice of bacterial antigen complexes. The cross-linking produces visible clumping, which can be seen with the naked eye or under a low-power microscope. So, principle of this coagulation, protein A of S. aureus bind Fc region of antibodies. Antibodies Fab region remains free to bind the antigen. Antigen bridges multiple antibody-coated bacteria, producing visible clumping. Clumping is equals to positive coagulation reaction, indicating the presence of the antigen in the sample. Next is other reaction. So other reaction, opsonization. Opsonization is a key immune defense mechanism where antibodies or complement proteins coat the surface of pathogen. This coating enhances the ability of phagocytic cells such as macrophages and neutrophils to recognize and engulf these pathogens. The Fc region of antibodies binds to Fc receptors on phagocyte, acting as a molecular handle for ingestion. Through this process, even bacteria that resist direct killing but become vulnerable to destruction inside immune cells. Opsonization bridges innate and adaptive immunity, ensuring efficient clearance of microbes from the body. This diagram, opsonization is a critical process in innate and adaptive immunity where pathogens are tagged for more efficient phagocytosis by immune cells like neutrophils and macrophages. It greatly enhances the ability of phagocytes to recognize and engulf microorganisms. So step 1, in the first part of this diagram, we see phagocyte like macrophages approaching bacteria. Phagocytes have some intrinsic ability to bind directly to bacteria via pattern recognition receptors, that is PRRS, that recognize microbial surface molecules. However, this binding is relatively weak and many bacteria can evade it. Second is complement mediated opsonization. Some bacteria activate the complement system. Complement proteins, particularly C3b, bind to the surface of the bacteria. Phagocyte expresses C3b receptors to CR1 on their surfaces. When bacteria are coated with C3b, The phagocyte can now recognize and bind the bacteria more effectively, leading to enhanced phagocytosis. So visual clue in this diagram is the bacteria are shown with C3b molecules attached and the phagocyte has C3b receptors engaging them. Next part is antibody mediated opsonization. So here some organisms do not activate complement effectively or complement activation is insufficient. In this case, antibody IgG bind to the pathogen surface. Phagocytes have Fc receptors that recognize the Fc portion of antibodies. This interaction allows the phagocyte to bind the antibody-coated pathogen and engulf it efficiently. Last part is combined opsonization by antibody and complement. So sometimes both mechanisms work together. Antibody binds the pathogen and antibody activates complement. So phagocytes now have both Fc receptors and C3b receptors engaged, which greatly enhances binding and phagocytosis. Next is neutralization. Neutralization is a protective immune reaction where antibodies block the harmful effect of toxins or viruses. For viral infection, antibodies bind to viral surface proteins and prevents the virus from attaching or to entering host cells, effectively stopping infection. In toxin neutralization, antibodies bind to bacterial toxins such as diphtheria, or tetanus toxin, preventing them from interacting with host cell receptors. These neutralizing antibodies form the basis of protective immunity acquired after vaccination or infection. Neutralization assays are therefore important for measuring vaccines' efficacy and immune protection levels in patients. Next is neutralization, this diagram. So neutralization is a process where antibodies inhibit the harmful effects of toxins or pathogens without necessarily killing them. This is a critical mechanism of humoral immunity, preventing toxins from binding to the target cells. So how this diagram works, toxin alone, toxin plus IgG or IgA, that is neutralization and its outcome. So free toxins, in this diagram, free toxins are shown approaching host cell, and without antibodies, the toxin bind to cell receptors and exert their harmful effect. Example is killing the cell or disrupting cellular functions. When toxin, that is neutralization, when antibodies are present, they bind to the toxin. Antibody binding blocks the receptor binding site of the toxin, preventing it from attaching to the host cell. So as a result, the toxin is neutralized. It cannot enter or damage the cell. So outcome, the host cell remain unharmed. This is the protective effect of neutralizing antibodies in both systemic immunity. This is also important for exam purpose. Next is complement fixation. Complement fixation is a classical serological reaction used to detect antigen-antibody interactions through the complement system. When a specific antigen and antibody combines, they fix and consume complement proteins from the reaction mixture. So in this test, if complement is fixed by the antigen and antibody reaction, it has no longer available to lyse indicator red blood cells coated with antibody. Thus, the absence of hemolysis indicate a positive result, while hemolysis remains, the complement was not fixed, indicating a negative result. Complement fixation tests were historically used for the diagnosis of viral and bacterial infection such as influenza, syphilis, and fungal infections. This is the diagram of complement fixation. So first step is serum plus antigen. Here, patient serum is mixed with specific antigen. If antibodies are present, immune complexes form. Next is complement binds to the addition of complement. So here, complement binds to the antigen antibody complexes. Complement is now fixed and unavailable for later reactions. Step 3. So if the complement was fixed, RBCs remain intact. that means no hemolysis positive test, antibody is present. This no hemolysis positive test, that means antibody is present. If complement was not fixed, that is RBC is lies, that is hemolysis occurs, and negative test, antibody is absent. So intact red cells positive complement fixation will give rise to patient has specific antibodies. Lysed red cells, that is equal to negative, patient lacks antibodies. Other complement-based tests, several other immunological assays used complement activity. The TPI test, that is Trypanosoma pallidum immobilization, is a confirmatory test for syphilis in which complement helps immobilize the pathogen when antibodies are present. Cytolytic assays measures complement-mediated lysis of cells, often used in immunological research to study immune cytotoxicity. Immune adherence test is based on the principle that complement-coated immune complexes adhere to specific cell such as erythrocytes or leukocytes, indicating complement activation. These tests highlights the diverse and powerful roles of the complement system in diagnostic immunology and research. Advanced immunological techniques. So, immunofluorescence is a technique where antibodies are labeled with fluorescent dyes to detect the presence of antigens in tissue or cells under a fluorescent microscope. In the direct method, the fluorescent dye is attached directly to the primary antibody, which binds specifically to the antigen, allowing immediate visualization of the antigen's location. In indirect method, an unlabeled primary antibody binds the antigen first, and then a fluorescently labeled secondary antibody binds to the primary one. This indirect approach amplifies the signal, making it more sensitive and versatile. Immunofluorescence provides both specificity and localization, allowing visualization of antigen distribution within cells and tissues in microbiology and pathology. This is the diagram for immunofluorescence. So, immunofluorescence is a powerful laboratory technique that uses antibodies tagged with a fluorescent dye such as fluorescein to detect the presence of specific antigen in cells or tissue sections. When viewed under UV light using a fluorescent microscope, the tagged antibodies glow, indicating when the antigen is located. Fluorescein, the fluorescent tag. So fluorescein is a fluorescent dye that can be chemically linked to antibodies. Under UV light, it emits bright green fluorescence. These allow scientists to visualize the precise location of the antigen in tissues or cells. So this A part, the fluorescent tagged antibody binds directly to the antigen, glows under UV light. One step, it is simple and direct method. This B, the primary antibody binds antigen, but the secondary fluorescent labeled antibody binds the primary antibody. Brighter signal, this is indirect immunofluorescence. C part, here complement is deposited on cells and a fluorescent labeled anti-complement antibody reveals the location of antigen antibody complement complexes. So emphasize fluorescent allow visualization of antigens or immune complexes precisely where they are in cells or tissues. Next is applications of immunofluorescence. Immunofluorescence is used in a wide range of clinical and research applications. It is a key method for detecting the rabies virus in brain tissues, providing rapid providing a rapid post-mortem confirmation of infection. It is used in the FDA ABS test, that is fluorescent trypanosome antibody absorption for the diagnosis of syphilis, detection of Toxoplasma gondii in clinical samples, also relies on fluorescent-based assays. In autoimmune diseases, indirect immunofluorescence is used to identify anti-nuclear antibodies, that is ANA test, in condition like systemic lupus erythematosus. Thus, immunofluorescence combines highly sensitivity and visual precision, making it indispensable in diagnostic laboratories worldwide. So, summary for this, Antigen antibody reaction overview. Antigen antibody reaction is the basis of immune recognition and diagnostic testing. Antigen contain epitopes and antibodies have paratopes that fit together like a lock and key structure. Their binding forms immune complexes which can be detected as visible reaction, that is precipitation, agglutination, complement fixation, etc. Role in immunity and diagnosis. In immunity, antibodies neutralize toxin, block viral entry, and opsonize pathogens for phagocytosis. In diagnostics, the same reaction detects antigens or antibodies in patient samples using ELISA, Western blot, and agglutination test. Historical background. Behring and Kitasato in early 1890s discovered antitoxin, that is the foundation of immunology. Landsteiner is antibody specificity and blood grouping. Merak in 1934 discovered lattice hypothesis for precipitation. Ria and Eliza will study this and next class. Modern highly sensitive immunoassays. Stages of antigen and antibody reaction. There are three stages: primary stage, secondary stage, and tertiary stage. In primary stage, we see antigen antibody binding, that is microscopic. Secondary stages, visible reaction can be seen, that is precipitation or agglutination. And tertiary stage, biological effects in vivo, that is complement activation, opsonization, and hypersensitivity. Titer concepts: antibody titer and antigen titration. In antibody titer, highest serum dilution will be showing visible reaction. Antigen titration determines optimal antigen concentration for accurate testing. Test accuracy based on sensitivity and specificity. Sensitivity, it is the ability to detect even small amount, and it will also give few false negative result. Specificity, it is the ability to detect only the intended target and few false positives will be seen. Precipitation reaction occurs between soluble antigens and antibodies forming insoluble precipitate governed by lattice hypothesis, that is cross-linking at equivalence zones. Zone phenomenon, there are three types of zone: prozone, equivalence, and post-zone. Prozone, antibody excess, here no visible reaction will be seen. Equivalence, optimal ratio, that means visible precipitation will be seen in this zone. And post-zone, antigen excess formation of antigen, that means again no visible reaction. Types of precipitation test: in liquid medium and in gel immunodiffusion. In liquid, we have seen three types of tests: ring test, Ascoli test for anthrax, flocculation test through VDRL test for syphilis, and tube test for quantitative measurements. In gel immunodiffusion, single diffusion in one dimension through Audin's procedure, double diffusion in one dimension through Oakley's procedure, and single diffusion in two dimension to radial immunodiffusion. Next, we studied about immunoelectrophoresis. It combines electrophoresis and immunodiffusion. It separates antigens by size and charge, then reacts with antibodies to form precipitating arcs. It is used to detect serum proteins, immunoglobulin disorders, and multiple myeloma. Next, we studied about rocket electrophoresis. It is quantitative immunoelectrophoresis. Antigen migrate through antibody containing gel under electric current, forming rocket-shaped precipitin peaks. Height of rocket is directly proportional to the antigen concentration. Next, we studied about agglutination reaction. It involves particulate antigens, example cells, bacteria, and latex particles. Types is direct agglutination, indirect, that is passive agglutination, and hemagglutination. Direct agglutination, natural antigen, for example, RBCs and bacteria. Indirect agglutination, it is soluble antigens coated onto carrier particles. Hemagglutination, it is RBCs clumping used in blood grouping and virology. Example is Vidal test, Brucella test, Will Felix test, and CRP latex test. Coombs test, that is antiglobulin test. There are two types of Coombs test: direct Coombs test and indirect Coombs test. Direct Coombs test detects antibodies already bound to RBCs, that is in vivo. Indirect Coombs test, it detects free antibodies in serum that can bind to RBCs in vitro. It is used for HDN transfusion testing, autoimmune hemolytic anemia. Next, we studied about opsonization. Opsonization is nothing but antibodies, that is IgG or complement C3b coat microbes, making them easier for phagocytes to ingest. Binding occurs through Fc region or C3b receptors on phagocytosis. Next, we studied about neutralization. In neutralization, antibodies neutralize toxin. Example is diphtheria and tetanus, or block viral entry into host cell. It is mediated mainly by IgG serum and IgA mucosal. Next, we studied about complement fixation, that is CFT. It detects antigen and antibody complexes by observing whether complement is fixed or free. No hemolysis is seen in this. Positive test, that means antibody is present. Hemolysis means negative test, that means no antibody is present. It is used in older serological tests for identification like influenza and syphilis. Next, we studied about immunofluorescence. It uses fluorescent dye tagged antibodies, that is fluorescin, to detect antigens. There are two types of immunofluorescence, direct and indirect. In indirect, labeled primary antibody binds to antigen, and in indirect, it is labeled to secondary antibodies binds to primary antibodies. Signal is amplified. Complement immunofluorescence, it detects complement deposition on cells. Where it is used in rabies detection, FDA, ABS test, ARN test for lupus, and toxoplasma diagnosis. For question part, for 3 mark, define an antigen antibody reaction. Second, what are epitopes and paratopes? Third, list 3 stages of antigen and antibody reaction. Fourth, state Marag's lattice hypothesis. Fifth, what is the equivalence zones in precipitation? Six, differentiate between sensitivity and specificity. Seven, give 2 examples of direct agglutination test. Eight, what is the principle of Coombs test? 9. Mention 2 clinical applications of immunofluorescence. 10. Define opsonization and name the main opsonins involved. For 5 mark question, first explain the stages of antigen antibody reaction with examples. Second, describe the lattice hypothesis and zone phenomenon in precipitation reaction. Third, compare direct and indirect immunodiffusion techniques with label diagrams. Fourth, explain the principle and application of immunoelectrophoresis. Fifth, describe the principle, procedure, and interpretation of the complement fixation test. 6. Differentiate between direct and indirect Coombs test with clinical uses. 7. Explain the mechanism of opsonization and its importance in immunity. 8. Discuss the principle and applications of rocket electrophoresis. 9. Explain neutralization reactions with examples of toxin and viral neutralization. And last 10, describe the direct and indirect immunofluorescent techniques and their diagnostic uses. So with this, we completed antigen and antibody reaction part. So in next class, we'll study about RIA, that is radio immunoassay, and ELISA, enzyme linked immunosorbent assay. So, sir, hello. So with this, we'll wind up today's class. So antigen and antibody reaction is completed.